Good afternoon. I'm Ganquan from the Center for Biosignatures Discovery Automation of VSU. It's a great honor to be here to present today's topic, the multiple sensor arrays for the single cells metabolic analysis. I will talk about 3 parts today. The first part is the background and the motivation. The next is the process flows of the fabrication. The last is the characterization of the device. So as we know, cells consume oxygen to acquire the energy, and at the same time, it produces hydrogen ions to the outside of the cell's environment. So the extracellular concentrations of oxygen and pH are the 2 key parameters for us to monitor the cell metabolism. And also, they are also the indicators of the disease state. For example, for the cancer cells, the cancer cell consumes less oxygen and generates more hydrogen ions comparing to the normal cells. So it requires particular devices for us to check the status of the cells. So here is a commercial product from the seahorse with the different sensors to detect the concentrations of the oxygen and the hydrogen ions for the bulk cells analysis. This process is carried out with a large populations of cells by measuring the parameters of the whole groups of the cells and the reporting average value. However, it's really hard to address the inhomogeneous cellular response to the external stimulus because the measurement of the cellular response is lost in the bulk average. For example, if there are 2 states, one state only has the pink cells, and another state has half red cells and half white cells. So obviously, the distribution of the 2 states are different, but they have the same average in terms of the cells color. It's impossible for us to distinguish between the 2 states in the population average. To reveal the actual mechanism requires the analyze at the single cell level. The single cell analysis allows the individual expressions studies, which is not limited by the averaging effect from the bulk population, and help to understand the behaviors of the individual cells when they interact with their local microenvironments. But the issues for the single cell analysis is if we go back to this page, if we assume that we put only 1 cell into this chamber, we could barely detect the change of the concentrations of oxygen and pH outside the single cell in such a large space. So this requires us to fabricate a micro chamber which is suitable for loading the single cell. And another issue is we cannot deposit the multiple sensors on a flat surface because the sensor solutions might mix to each other and affect the final result. So this leads us to make a multiple position for deposit the multiple sensors like this structure. So our goal is to use the method of the dual depth wet etching to fabricate multiple sensor trapping lead arrays, which each array has the multiple pockets for the multiple sensors. This video is a demonstration of fabricating the top lead arrays in the class 100 clean room. So at the beginning, we use the RC cleaning to remove, at the beginning, we use the acetylene to remove all the organic particles, and then we deposit morphosilicon, very thin layers. And after this, we after this, we spin coat the photoresist on its top, and we use the basic photolithography method to make patterns for the photoresist. And then by the help of the dry etch, by the help of the dry etch, we'll transfer the patterns from the photoresist to the morphosilicon. And if we move the photoresist, we'll see the morphosilicon wafer will serve as a hard mask. Then we put this device into the HF solution and do the first time wet etch. And the glass will have the first layers of the patterns. So when we reach this step, we are going to do another process, which is the second time wet etch. But before the second time, we have to still use the similar process. I mean the photolithography method. So we spin coat the photoresist again on this device, and then after exposing development, we'll have and with the proper alignment, the second layers of patterns will deposit on the left amorphous silicon, as shown here. Yeah. So the photoresist will use it as a soft mask to dry etch the amorphous silicon wafer, the amorphous silicon layer. Yes. And when the patterns from the photoresist transferred to the left amorphous silicon layers, we remove the photoresist, and then use it as the hard mask, and then do the second time wet etch. So if we remove all the left amorphous silicon, we'll have a structure like this. It has a circular lid, and inside the lid we have two pockets for the deposition of sensors. So we will drip the sensor solutions into each well and put the device into the oven, curing for overnight. So the sensors will be polymerized into each pocket, and then we'll use it for the assembly. So when the top lid is ready, we use the similar method to fabricate the bottom wells, but only use one-time wet etch. So we coat up thin layers of perlin-C and then load the cells into each well. We flip the top lid over, and if we look at the cross section, so we'll see. And if we place the top lid on the bottom wells, we'll see the cells and the sensors is placed in an enclosed space. And then we use the piston to press the top lid to make a sealed environment. And we'll use this device for the following test. So this on the left is the 5x5 arrays we designed. The pitch is 300 micrometers, and the diameters of each pocket is 70. And on the top right is the 3D images from the optical profiler. And at the top is the scanning electron microscope images to show the cross-section views of the microstructure. For the dimensions of the measurements, the depth of the pockets and the height of the lid we want to achieve is 20 micrometers and 10 micrometers. And the actual size we get after the fabrication is the 19.1 micrometers for the pocket and the 9.5 micrometer height for the lid. And similarly, for the bottom wells, we want the depth of the microwells to be 10 micrometers. And the final result is 10.6. As I talked earlier, if we want to get accurate detection of the parameters, it's critical to have the same microchamber volume for the metabolic experiments. That means the etch uniformity across the whole wafer is one of the most important parameters for the wet etch process development. We measured the height of the lid across the central section of a 4-inch wafer and plotted this result. We can see the lids located at the edge of the wafer were slightly higher than those located at the center of the wafer, indicating that the etch rate is higher at the edge. But the difference is only about 0.1 micrometers for 10 micrometer etch, which result in negligible variations. So this is the oxygen sensors and the pH sensor were polymerized into the pockets, and the oxygen. And we put the device into the buffer and then test the properties of the sensors. So the oxygen sensors responded a strong signal when the sensor is put into the environment which has lower concentrations of oxygen. And similarly for the pH sensor, it reflected strong signals when the buffer. When the pH value of the buffer is above 8. So according to this property, if we put these sensors or this device for the single cell analysis, if the cell is consuming oxygen, and the signals of the oxygen sensor will increase. Also, if the cells produces the hydrogen ions, the signals of the pH sensors will also increase. So this gives us the basic concept, the result of how the cells, the oxygen consumption of the cells, and also indicating if the cells is a cancer cell or a normal cell. So in summary, we successfully fabricate the dual sensor arrays, and also we will continue the single cell metabolism experiments, which will monitor the cells about the change of the oxygen concentrations and the pH. So at the last, I want to thank Dr. Meldrum, who is the director of our center, and also all the faculties and students who helped me a lot to finish this project. And also thank Caesar for providing me the clean room environment to fabricate this device, and also NIH for the funding, and ASU, GPSA, the travel grant for me to be here. Thank you.